art philosophical section, I start with a quote by John Dryden in a poem from 1681 that self-defense is nature's oldest law. So we've all received from our ancestors all the genes that they needed to survive every pandemic, every famine, every hard winter, at least until they had their first child. So those are the genes that we've inherited, okay? But the environment's changed. And so, you know, those genes are not enough. Another quote that I like is from Nelson Mandela, and he says that when a deep injury is done to us, we can never heal until we forgive. And then you know, I was talking to Keith last night, and he asked this similar question: how does a cell decide when to forgive? Obviously, that's a metaphor, and begin to heal. And so the answer is a cell will only redirect resources, you know, and those resources are both energetic and material resources, so matter and energy and information, away from cellular defense toward healing when it receives the signals that say the future is safe. And the corollary is that regeneration and healing can't proceed until persistent triggers of the cell danger response are removed. Okay, like if you keep ripping the scab off of an abrasion, it's going to take you much longer to heal, and you're going to have scarring. Okay, if that keeps happening. So then this big question, overarching question: why do people become chronically ill? Well, I believe that that's because they have encountered a perfect storm of usually dozens of environmental and genetic factors that produce or persistently reactivate an injury and then block the normal cycle of healing. And the corollary is that all chronic illness represents a failure of our ancestral genes and our inherited epigenetic modifications to adequately manage the response to environmental conditions that we've experienced. And so basically it leads to this logical tautology. People are sick because they can't get better, okay? Like, you know, so why can't they get better? Well, you know, I believe it's that the information is in the molecular analysis of the healing process itself. So the organizing principles of our research start out with this one fundamental fact: injury is universal. Living cells try to heal themselves, and cells that can't heal cannot transmit their DNA to the next generation. So they don't, and they die, and those genes, that gene set dies out. Chronic illnesses are the result of a mismatch between genes, environment, and the developmental timing of the injury exposure. So gene by environment by timing, or we refer to this as epigenetics. And then finally, the last organizing principle that Chauvin has iconized, and that is metabolism is the real-time interaction of genes and environment. So our genes give us a certain plasticity to respond to environmental insults, and so metabolism lives in the moment. And I'll follow this up by saying metabolism is fractal. There are it's like an ancient oak tree where there are metabolites that are trunk metabolites that don't change at all. There are very thick branches, and then there are leaves that flutter in the wind, and even those can be remarkably useful in understanding disease. For example, one of the metabolites that flutters in the wind like a leaf is glucose. But we know that fasting glucose can be very useful in looking at someone's insulin resistance or sensitivity. Okay, so that's for you, Chauvin, down at the bottom right. Okay, so the take-home messages are that pathogenesis does not equal salogenesis, and I'll define that word, but it's basically the process of injury does not equal the process of healing. This is really fundamental and absolutely a missing piece of medical approach to disease that we all were trained in. So the cell danger response and mitochondria coordinate both of these processes, and metabolomics can be used for both pre-symptomatic risk assessment triage, diagnosis, and prognosis. Okay, so where does this leave us? So we cannot see what we cannot imagine. Well, I won't go, this will take too long if I go into too many analogies, but I'll say our training creates subliminal limits or unrecognized horizons in our vision so that, new tools and models, can be used to extend our scientific vision. So sometimes in order to see, a new world, all you need is to create a new lens or a microscope or a telescope or a mass spectrometer. So a paradigm shift is needed to understand chronic illness. I'll say this many times throughout the talk, acute disease is not the same as chronic disease. You cannot use the same principles and hope to get the same results. So I consider acute disease the medicine in book 1 is the last 5,000 years of recorded human history, where we really focused on managing what happens if you break a bone, trauma, or poisoning, or if you have infections. So medicine's gotten good at this. But book 2 of medicine is really anything that persists for longer than 6 months. Okay. All right, so imagine a vibrantly healthy human as, this house on the left before disease. Well, you can suffer injury, and there are mechanisms of injury. In this case, it's a fire. That is my metaphor for pneumococcal pneumonia or a viral encephalitis. Okay, we are trained to identify the mechanisms of injury and then to treat those. Okay, so we can treat that fire, but then what we're left with is a house that is destroyed. Now, all of first book medicine stops with step 3 and then relies on spontaneous healing to occur. We do this so subconsciously that it's automatic that if we give, let's say penicillin for a pneumococcal pneumonia, we just assume that all the pulmonary, destruction that has occurred as part of the infection will heal itself. Okay, and we forget that's an active process. So rebuilding requires active healing, or what I call salogenesis. And that is what I believe is a critically missing component of modern medicine. So Salus was the Roman goddess of personal health, welfare, and safety. This slide starts with basically saying that healing is not the reverse of pathogenesis. So we can start with health. You can suffer a traumatic injury, be exposed to toxic metals or pollution or viruses, tobacco, infections of different kinds, and get over to the right square, this red square, which is a chronic illness. But it turns out, so we can study that. And what first book medicine teaches us is we just eliminate those problems and everything will get better. Okay, that's what first book, but that's not how living systems work. That's how you repair a damaged or imperfect Hubble telescope, is you analyze its optical properties, you create a fix, you fly it up, install it, and then you enjoy the fruits of your engineering fix. That's not how cells repair themselves, okay? So there's a beginning and a middle and an end to this process of returning back to health that requires transformation of mitochondria from different developmental phenotypes, what I call M1, M0, and M2 mitochondria, okay? So in this first book of medicine, treatments of chronic illnesses will lead to palliation. So they're usually symptomatically based. So if someone has a headache, you can give them an aspirin, or you can, remove the hammer that keeps hitting them. Okay, so based on the word salus, we've used this word salogenesis to describe the molecular and cellular events that are necessary for going from a state of injury to a state of health. And that's what I call second book medicine. And what I believe that treatments that really focus on this second book on healing actually leverage the internal ability of cells to use information is stored in our DNA and as well as energy produced by our cells and material resources in the form of amino acids and nucleotides and fatty acids and sugars. Okay. So I gave this talk in April of last year at NIH, and the organizer that invited me, said, "Great talk, Dr. Naviaux, but I hope you're wrong." And I said, "Well, why is that?" Well, if you're right, it means that the National Institutes of Health has been funding only the first limb of a two-limb problem. We've only really been funding, understanding the pathogenesis of illness and not the sallogenesis. So for 70 years almost, the NIH has really focused on building deep silos of knowledge for many, many different diseases, for primary mitochondrial disease, autism, chronic fatigue syndrome, Gulf War. I put these silos here because these are the silos. Well, 10 of these are ones that we've already studied, and ALS, Parkinson, and Alzheimer are ones that we will be studying over the next few years. And so what people, by building these deep silos of knowledge, have done is they have focused on the differences between illnesses. And what we are focusing on is the similarities, this hidden, the commonality that all diseases share is in order to get back to health, there is an underlying root to the process of recovery. So the things that make chronic diseases the same are more important for healing than their differences. So this has, political and social implications for our unrest in America at the moment. But really, it's our similarities that are more important for healing than our differences. So how did I get into this? Well, I got into it because of patients with primary forms of mitochondrial disease, genetic forms of mitochondrial disease. So these are diseases called MELAS and NARP and Alpers syndrome. We actually discovered the gene that causes causes Alpers syndrome, which is the oldest Mendelian form of mitochondrial disease, first described in 1931. But anyway, so under health conditions, mitochondria are a reticulum. They are almost a root system that go throughout the cytoplasm of a cell. So here are two HeLa cells next to one another down at the bottom with an interconnected network of mitochondria. But when there's any kind of injury, mitochondria fragment and they change their metabolism and they become pro-inflammatory. Okay, so these are anti-inflammatory organelles that are consuming oxygen. These are pro-inflammatory organelles that are no longer consuming oxygen efficiently. When health is restored, the mitochondrial reticulum is restored. They're reconnected. But when that's blocked, they remain disconnected. This disconnection has been seen also even on the space station. When you grow cells in microgravity, mitochondrial would become disconnected. So anyway, when this happens, when you're blocked to healing, 60% of adults in the world today suffer with at least one chronic illness, and actually 40% of children, in their up through their teens, suffer with at least one chronic illness. So we've conducted over a dozen different studies, 2 in autism and 2 in chronic fatigue syndrome, but also Gulf War illness and post-traumatic stress disorder, traumatic brain injury, recurrent major depressive disorder with and without suicidal ideation, an autoimmune liver disease called primary sclerosing cholangitis. We've studied exercise and aging and these primary mitochondrial diseases, and then a longevity default pathway in C. elegans called Dower, and some breast milk biomarkers that I'll talk about. Well, where does that all lead us? It leads us to what we call the cell danger response. So the core pathways of the cell danger response are regulated in over a dozen chronic illnesses that we've studied. And so if you think of measuring metabolites, a set of metabolites that are unique for a particular disease, as well as metabolites that are shared and pathways specifically that are shared by different diseases, what you come up with is something that looks like a sunflower, where the shared pathways are the pathways of the cell danger response. And those involve purines and pyrimidines, phospholipids, acylcarnitines, fatty acids, sphingolipids, eicosanoids and endocannabinoids, cholesterol, folate, B12 metabolism. I can go through it, but basically all these are part of a system of cogs and wheels that are coordinately regulated. These are not things that are activated in isolation. They do not work in isolation. They work as an integrated system. So what is the cell danger response? So here's a cartoon of a cell. An archetypal stress was a viral infection. And one of the first things that happens is, I'll point your attention to number 4 over here, is actually a conversion of filamentous mitochondria to segmented mitochondria. We call this the spaghetti to meatball transformation. Okay, so this is, and that's actually not just a joke, it's what the mitochondriacs call it. Okay, so the spaghetti to meatball transformation. But when that happens, then the intracellular dissolved oxygen concentration rises. When that happens, then NADPH oxidases are activated, and superoxide and hydrogen peroxide are created, and you get a host of many other things. So this is an ontogenetic response, meaning that it's a developmental as well as genetic response initiated by what I call electron steals, because mitochondria regulate their metabolism according to the electrons they receive from metabolism. When those electrons are stolen by a virus, let's say, so to make its own proteins, its own lipids for cell envelopes, or nucleic acids, then there's fewer of those that are available to mitochondria, and that results in this filamentous to fragmented morphological transformation. And so I'll refer you to the paper. We can go through, I won't go through everything here, but the important thing is that this is a coordinated response, but people tend to look at this through a keyhole of their own research interests. So some people might just look at sphingolipids. Other people might look at cholesterol or fatty acids. Some people might look at, demethylation of DNA. Some people might look at endogenous retroviral sequence activation. Okay, these are all coordinately regulated responses. All right, so again, to nail this home, so we start in a condition of health. This is first book of medicine, acute illness. We have an injury. Doctors will look after the airway, breathing, and circulation. You do first aid, you provide symptomatic treatment, and because this black box is intact, most of the time people get better. But if the black box of healing is not, getting better, then it tips us into this repeating loop of chronic illness. Okay, so now if we just start with this as a dysfunction as a problem, as a starting assumption in all chronic illness that results from a variety of different problems that are relevant either for children in psychosocial deprivation states or rewounding in adults, it'll pull us into this repeating loop. Okay, but if we unpack that black box and start analyzing it with the tools of systems biology, we can begin to see that there's actually an organized sequence of events that happen. And I call these the cell danger response 1, 2, and 3. And we'll go through those a little later. But if you unblock that, then healing is restored and health can be restored. So this is such an ancient response. Plants have it too. So this is a Arabidopsis. This is a little cricket bite over here. Turns out if you just add glutamate or ATP to this, this genetically modified Arabidopsis that will fluoresce green in the presence of uncaged calcium, this is what happens. Does everyone see that? So that's the plant within 90 seconds transmits the injury that it receives in one leaf to all parts of the plant. Okay? Humans are even better at doing this. Okay, so we spend most of our life in this health cycle of wakeful activity and nutrient intake. That activity releases a little bit of extracellular ATP that then has to get metabolized to adenosine, which is necessary for the induction of sleep. If you don't have adenosine, you can't have normal restorative sleep. Okay? Once that's metabolized, then you can wake up again. So, if the injury is very significant, let's say with a viral infection and there's been cell loss or excitotoxicity, cell loss, then the first stage of the cell danger response is activated. That's inflammation. It's like trying to seal off the bulkheads of a submarine that has been injured, so that the rest of the submarine will not be destroyed when one compartment becomes filled. So that's what this containment and innate immunity is all about. And there's a very specific type of metabolism called glycolysis that is used during that time. Let's see. So, and this can be activated by any number of different things. And so I have a stimulated emission video of mitochondria after they've sustained a stress. And within 5 minutes, they transform from the spaghetti to meatball configuration. That changes them from antioxidative, basically anti-inflammatory to pro-inflammatory organelles. All right. So these spherical mitochondria we call the M1 mitochondria. M0s are pluripotential. They can actually be both differentiate into M1 or M2 mitochondria. And M2 are the more filamentous oxphos dependent forms of mitochondria. Okay. So these are just the very specific stages of metabolism that always exist. It doesn't matter what the injury is, whether it's a broken leg, a cut, a scrape, a myocardial infarction, or a stroke. So now is the second half of the talk. So